Quantitative MRI of the spinal cord, reproducibility and normative values across 40 sites. My name is Alonso-Ortiz, and I have no financial interests or relationships to disclose with regard to the subject matter of this presentation. Quantitative spinal cord MRI is challenging. It's challenging for a number of reasons. Firstly, quantitative MRI methods have historically been developed for the brain, and in order to apply some of these methods to the spinal cord, they need to be adapted and optimized to meet the unique challenges that are presented in the spinal cord region, such as its small size, its mobility, and the fact that it's surrounded by tissues and organs that have different magnetic susceptibility. So many of these challenges are being addressed. However, there's an important issue that remains, and that is that there's no clear consensus for acquiring quantitative MRI data in the spinal cord. This means that a lot of time and money is lost setting up scans every time researchers want to acquire quantitative spinal cord images, and this can also lead to large variability in multi-site, multi-vendor data, which is of course detrimental for assessing biomarkers, what we're interested in. So in order to solve this problem, we put together a consortium of international spinal cord researchers with the goal of developing a consensus protocol for quantitative spinal cord imaging at 3 Tesla with Siemens, GE, and Philips scanners. The protocol is publicly available and includes a standard operating procedure, SOP, which describes the acquisition instructions and important tips, such as how to center the field of view, how to position slices, how to adjust the shimming box, etc. The protocol includes a number of contrasts that can be used for quantitative assessment of spinal cord impairment, such as T1 weighted imaging, which can be used for measuring the spinal cord cross-sectional area, CSA, T2 weighted imaging, which is also used for CSA measurement. CSA has been shown to be useful for assessing atrophy in the spinal cord, so that's why it was included. Diffusion weighted imaging is included for diffusion tensor imaging, which provides metrics that are sensitive to demyelination and axon degeneration. Magnetization transfer imaging is included as well. The magnetization transfer ratio is also sensitive to demyelination. And lastly, multi-echo gradient echo imaging is included for measuring CSA and gray matter, a metric that's useful for assessing gray matter atrophy in the spinal cord. So 40 sites acquired data in healthy adults using this protocol, and the acquired data is also publicly available on GitHub. So today I'd like to show you the results that we obtained when analyzing that data. And so the objectives of this talk are to present the quantitative MRI biomarkers for spinal cord impairments that we derived from the multi-site dataset using the consensus protocol. These metrics are spinal cord and gray matter CSA, fractional anisotropy from DTI, and magnetization transfer ratios. So now back to the multi-site data. Out of the 40 sites that participated in data collection, 6 had GE scanners, 8 had Philips scanners, and 26 had Siemens scanners. Each site collected data in 6 participants for a total of 248 data sets, with all data sets including T1 weighted, T2 weighted, diffusion weighted, MT, and multi-gradient echo images. The datasets were processed using the Spinal Cord Toolbox, and the analysis scripts are also publicly available on Read the Docs. The website includes text and video instructions and tutorials for how to process the data. This figure here shows the full processing pipeline, and briefly to go over it, the spinal cord was segmented on the T1 weighted scan, registered to the PAM50 template, and the cord CSA was computed for each slice. For all the other contrasts, the spinal cord was segmented and registered to the PAM50 template using the transformation obtained from the T1 weighted scan. The cord CSA was also computed from the T2 weighted scan, gray matter CSA from the multi-echo gradient echo scan, white matter MTR from the MT weighted data, and white matter FA from the diffusion weighted data. So next, intra and intersite coefficients of variation were computed. And without further ado, here are results for the CSA obtained from the T1 weighted scans. The bars represent the average CSA between cervical levels 2 and 3, so that is C2 and C3 for each site, and with the individual subject values shown as the red points. The mean and standard deviation are also shown for each vendor as a dashed line with a shaded window around it. And the intra-site COVs were averaged for each vendor and found to be all under just about 8%. And the inter-site COVs for each vendor ranged from about 3% to 4%. Here we have the CSA obtained from the T2 weighted scans. Again, intra-site COVs are close to 8%, and inter-site COVs ranged from 3.5 to 5.6%. You may have noticed at this point that T2 weighted images resulted in slightly larger cord CSAs than T1 weighted images. So in fact, we found that there was a significant linear trend between T1 weighted and T2 weighted cord CSAs for all three vendors. And we believe that researchers can use these trends to account for differences in T1 weighted versus T2 weighted cord CSAs in their data. Now on to the gray matter CSA, which was averaged across C3 and C4. Here we saw higher intra and inter-site COVs in Philips scanners, and we believe that this is likely due to the fact that some sites used older versions of the consensus protocol, which produced lower contrast between white and gray matter, and as a result, less reliable gray matter segmentations. Our MTR values were averaged between C2 and C5. Magnetization ratios were comparable between Philips and Siemens scanners, with GE results being somewhat higher. This is likely due to the fact that a different MT pulse is used on that platform. And lastly, here are our results for FA, which was averaged between C2 and C5. Intra-site COVs ranged from about 4% for Siemens scanners to 12% for GE scanners, while inter-site COVs ranged from 3% for Siemens to about 6% for GE. So in general, we observed that intra-site COVs were roughly twice that of inter-site COVs, and this tells us that subject variability largely accounts for the differences that we observe across the sites. On average, COVs were around 5%, which in turn indicates that the implementation of the spinal cord consensus protocol was successful. So overall, our data show that the spinal cord consensus protocol is reproducible across sites and vendors, and we hope that it can serve as a reference guide for clinicians and researchers that are interested in quantitative spinal cord imaging, but may not have the technical background that's required for protocol optimization. We're living in a time in which many believe that science is experiencing a reproducibility crisis. With our open access, multi-center data set, our open source analysis pipeline, and our publicly available consensus protocol and SOP, we hope that we can enable more transparency and reproducibility, as well as facilitate multi-center quantitative spinal cord MRI studies.